In vivo CAR-T had one of its busiest stretches in months. First patient dosing, new primate data, ASGCT talks everywhere. And if you only remember one thing, make it this. The field is not waiting on a better acronym. It is waiting on boring proof that the right cells got programmed for the right amount of time in actual humans. So this is a weekly brief on recent in vivo CAR-T, in vivo immune cell programming, and adjacent immune engineering news. The date on this one is May 25th, 2026. The angle is not that everything is solved now. The angle is that 4 updates matter, but they matter for different reasons, and people keep flattening them into the same story. First, the actual human milestone. On April 23, 2026, Create Medicine said it dosed the first patient in a phase 1/2 study of MT-304. That is important because it is real clinical movement, not another mouse victory parade. But there is an equally important caveat. MT-304 is not a standard in vivo CAR-T story. Create describes it as a HER2-targeted multi-immune in vivo CAR therapy that programs NK and myeloid cells using an mRNA LNP system. So yes, it belongs in this lane. No, it does not mean we suddenly have clean human efficacy data for in vivo T-cell programming in solid tumors. We do not. Second, the most useful preclinical update for understanding where the field is going. On May 8, 2026, Azalea announced late-breaking ASGCT data showing first-in-primate in vivo TRAC-CAR T engineering. Their pitch is very specific. Put the CAR into a defined genomic locus, the TRAC locus, so expression is controlled by the cell's own machinery instead of just floating around as a generic transgene. In 6 out of 6 rhesus macaques, they reported deep B-cell depletion in peripheral blood by day 10, plus strong activity in lymph nodes and bone marrow without lymphodepletion. Why does that matter? Because Azalea is making the argument that precision integration is not a luxury feature. It may be a safety and durability feature. That is a serious idea. It is also still primate data. If you hear first in primate and your brain auto-completes to clinically solved, please put that brain on a timeout. Third, Sana's SG293 update is less flashy than the Azalea headline, but may be more practical to watch near term. Sana's April 27th ASGCT announcement framed SG293 as a CD19-directed in vivo CAR-T candidate using a CD8-targeted fusion-based delivery system. The company emphasized cell specificity and avoiding potentially troublesome delivery to tissues like liver. That is the correct fight. In vivo CAR-T is not just can you make a CAR, it is can you avoid programming the wrong tissues on the way there. Sana's own SEC filing from March 3, 2026 also matters here. It said a surrogate for SG293 generated robust in vivo CAR-T expansion and deep B-cell depletion in cynomolgus macaques after a single IV dose without lymphodepletion. Again, encouraging, still preclinical, still a surrogate system, still not the same thing as durable human benefit.
Fourth, Kernal Biologics brought a slightly different answer to the same problem at ASGCT. Their April 30 release pointed to T-cell-targeted lipid nanoparticles, plus what they call selective translation. In the ASTCT abstract publication, the pitch is basically a double lock: targeted delivery to CD8 cells, then an RNA architecture meant to keep payload expression from turning on in the wrong cells, like B cells or hepatocytes. That is a very revealing theme for the whole field. Everybody now understands that delivery alone is not enough. The platform also needs a second control layer. And this is where current coverage gets sloppy. A lot of it sounds like body as bioreactor, factory-free CAR-T, simple infusion, democratized access. Some of that may become true, but the more honest summary right now is that the field is experimenting with different control stacks: viral versus non-viral, transient versus integrating, T-cell only versus multi-immune programming, cell targeting plus selective expression, or cell targeting plus site-specific integration. The real competition is not just potency, it is controllability. Now, let me give you the practical viewer filter I wish more people used. When a company says in vivo CAR-T or in vivo immune programming, ask 3 boring questions. Question 1, who actually got programmed? Endogenous CD8 T cells, NK cells, myeloid cells, a mixed population? If the answer is fuzzy, the headline is ahead of the biology. Question 2, how durable is the programming supposed to be? Transient expression can look safer, but it may need redosing. Integrating approaches may help persistence, but they raise a different safety and manufacturing conversation. There is no free lunch here, which is rude, but very on brand for immunology. Question 3. What species and stage is the evidence in? Mouse data is useful. Primate data is more translationally interesting. Human data changes the conversation. These are not interchangeable proof tiers. This is also why I think the most important context story is still Kelonia's early human KLN 1010 data from ASH 2025. That was one of the first genuine signs that a single off-the-shelf in vivo CAR-T infusion could generate meaningful anti-BCMA CAR-T expansion in patients with multiple myeloma without lymphodepleting chemotherapy. The numbers were tiny and early, but it moved the field from "is this even biologically possible" to "which delivery control strategy will survive contact with humans?" What are people missing right now? 2 things. First, off-the-shelf is a logistics claim, not an efficacy claim. You can absolutely improve access and still fail biologically. Second, adjacent immune programming may matter as much as classic T-cell programming. Create's first patient MT304 update is a good reminder that some companies are trying to solve solid tumors by programming NK and myeloid biology, not just by making a better T-cell missile and hoping the tumor microenvironment behaves. What could fail from here? Plenty. Off-target delivery, transient activity that is too weak, persistence that is too durable in the wrong way, redosing problems from immune recognition, solid tumor trafficking issues, safety differences that look fine in macaques and less fine in people, and maybe my favorite recurring biotech trick, people confusing a platform milestone with a clinical outcome.
So, what should you watch next? Watch for actual human safety and pharmacodynamic readouts, not just prettier platform diagrams. Watch whether lymphodepletion-free approaches still generate enough activity in patients. Watch whether companies can show tissue specificity, not only blood readouts. And watch whether the autoimmune and solid tumor branches of this field start separating, because they may reward very different delivery designs. My opinionated wrap-up is this. The field did have a meaningful week, but the practical behavior change is not pick your favorite company and clap when it says in vivo. It is grade every claim by control layer and proof tier. The headline is "In vivo CAR-T is getting crowded." The real story is that delivery decides, and human proof is still precious.